Hello my lovely friends, I am Dr. Arun Swami. In this video we'll discuss about the mechanism of skeletal muscle contraction and also we discuss about the sarcomere. So first we should understand that what is the sarcomere and later we will discuss about the myocyte contraction. The sarcomere is the structural and the functional unit of the skeletal muscle which extends from one Z line to the next Z line. So if you will see to the muscle, the skeletal muscle is actually look something like this. In this skeletal muscle, if we take the small portion, it will look something like this part, and then if you will see the another zoom section inside of this muscle, it will look something like this. And this is part which we call as myofibril. In this myofibril, if you will see little bit more closer look, more zoom section area, guys, what you can see that there are some lines are seen over here. These are actually the Z line. These are actually the Z line. Now, one Z line on this side, another Z line on this side. So these are the Z line. Now, in between, there is another line we call as a M line. So what you can see that this zone from this Z line to this another Z line, this complete area which you seeing over here, this we call as sarcomere. And this sarcomere is structural and the functional unit of the skeletal muscle, and the muscle we very well know that this required for the contraction. So how the muscle is actually contract, what are the different protein inside of this skeletal muscle, we will discuss about in this video. So you can see first of all, here you are seeing that if this is your sarcomere, you should do the more zoom area to this sarcomere. Once you will do the more zoom or more that highlight area of this sarcomere, what you can see that this sarcomere will look something like this. In this sarcomere, you can see there is some blue lines are there, and then there are some red color lines are there. Actually these are the proteins, because we know inside the body each and everything is mainly made up of the protein. So they are actually the protein which is called as a contractile protein. This yellow color protein which you can see over here, this is your actin, and this red color protein actually your myosin. And guys, please pay attention, this actin and myosins are your contractile protein which helps to do the muscle contraction. There is another protein which you can see looking like a spring like protein. This is we called as titin. And guys, very important part, this titin is actually the elastic protein which provide the elasticity of the muscle. And if this titin is going to be absent or mutated, guys, the elasticity of the muscle get lost, and that's the condition happens in the heart muscle. When the heart muscle lost the titin protein, guys, there is a condition called as dilated cardiomyopathy. So this titin is very important for provide the elasticity of the muscle. Now if we talk about how the muscle gets contract, so the muscle is actually required essential ion to contract, and that is your calcium ion. Guys, please pay attention, calcium is not only for the bones, calcium is actually mainly required for the muscle contraction, and muscle contraction provides day-to-day locomotion for us. So bones are actually simply bank of the calcium, but the main calcium is required for the muscle contraction. Now please pay attention how this muscle is actually contract. Out of this two protein, myosin is thick and actin is thin, and you can see that this myosin are having some projections over here. These are we called as, these projections are actually we called as myosin head. What we call this? These we called as myosin head. Guys, these heads contain a special structure over here we called as ATP, and we know the ATP is actually the power of the body, and this ATP helps for the muscle contraction. Whenever muscle needs to be contract, this myosin head get attached with the actin binding area and helping with the ATP. They will tilt like this from this side, and they will tilt like this from this side to this side, and leading to this tilt, this muscle gets contract. Whenever muscle is getting contract, guys, the size of sarcomere will be shortened. Now please pay attention. There are some special bands into this sarcomere, which we call as I band, A band, or also we call as H zone. Guys, please pay attention. The area where this myosin and the actin is actually present, this area we call as A band. But the area where is only and only the actin is here, and in another sarcomere here is only the actin is here, combinedly these both actin area we called as I band. Similarly on this side, this actin for this sarcomere, this actin for this sarcomere, and this combinedly area we called as I band. So you can see over here that this side is I band, this side is also I band, but middle portion we called as A band. Guys, please pay attention. In between area where this actin is absent, only and only the myosin is present. Guys, this area we called as H zone. What we call this? We called as H zone. H zone. So very, very important. There are special bands, I band, A band, and the H zone. Guys, please pay attention. Whenever muscle gets contract, whenever muscle is getting contract, the size of sarcomere is decrease. During this muscle contraction, what happens with these bands? Guys, please pay attention. Very, very important MCQ in several examinations like in FMGE or in the NEET PG. Guys, whenever this sarcomere will be contract, the size of I band will be decrease. This size is also decrease for this side of the I band, but the size of A band, it will remain constant because only the myosin head is tilting. Itself myosin is not moving, only head is tilting. And guys, suppose guys, if this is the myosin, this is the head, only head is moving. So size of myosin is always remain constant during the muscle contraction. Repeatedly asked MCQ in various examination. But also, what happened with the H zone? Guys, whenever the muscle is getting contract, this actin and this actin, it's coming closer, and the size of H zone is also decreased, and in severe contraction the H zone can be disappear. So one more time, this is your sarcomere, which is having the border lines which we called as Z line. Then there is a middle line we called as M line. There are two bands, I band and A band. The I band is only the actin area and A band is actin and myosin area. This is your A band, and the H zone is only the myosin containing area, and this is present into the middle of the sarcomere. So this is your sarcomere, very, very important structure. Sometimes examiners give you that how this sarcomere looks like on the electron micrograph. So this will actually look something like this. And in the various examination, they used to ask that they will put a arrow and they will ask what is this structure, what is this structure, what is this, or what is this. So if you will see, if you will compare with this image, you can easily understand that this is your Z line, then this is your M line, then this is the dark zone. This dark zone is actually your A band. This dark zone is your A band. This dark zone is also the A band, and this middle zone is your H zone. This middle zone is your H zone, and this is your I band. What is this? I band, and this is also your I band. So I hope now nobody will forget how this sarcomere it actually looks on electron micrograph, right guys? So please pay attention. The things just now we discussed, the bands and the lines. The A band, which is the dark in color, that is the length of the thick myosin filament, which is always constant during the muscle contraction. I band, which is light band, which is thin actin filament only, and this shortens on the contraction. H zone, that is the central clear area in A band, which is disappears during the contraction, and M line is center of the sarcomere, and Z line, that is the boundary of the sarcomere. And now this during the contraction, sarcomere length will be decreased, I band and H zone will be shortened, but A band will remain constant, and there is an overlap between actin and myosin increases. The protein just now which we'll discuss, actin, which is the thin filament, and it lives with the actin, tropomyosin and troponin. Troponin is having the three binding site, that is troponin T, troponin I and troponin C. Myosin is thick filament which is contain the ATPase and it required for the cross bridge formation. So whenever this myosin had attached with the actin, it is making like a bridge. So this is we called as cross bridge formation, and titin is the elastic protein from Z line to M line. Now how this muscle is actually contract. So if we'll talk about, earlier we discussed sarcomere, but now we are seeing complete skeletal muscle cell which we called as skeletal myocyte. Please pay attention. In skeletal myocyte, if you will see the different structures are there, there are some depression over here. These depression we call as T tubules. On this T tubules, there is a DHPR receptor which stands for dihydropyridine receptor, and there is another receptor present which is DHPR is attached, we call as RYR, ryanodine receptor. Guys, there is a storage house of the calcium inside this muscle cell, and this storage house we called as sarcoplasmic endoplasmic reticulum, which is actually the calcium storage area inside the muscle cell. Because we know if the muscle need to be contract, muscle required the calcium, and we also understand that the calcium only should be present inside a closed area, because if the free calcium will remain inside of this muscle cell, guys, without any message of the brain, this muscle get easily contract. But we know muscle should get always contract with our willingness, and we gives the message via the brain. So whenever the brain will send the message, then only this muscle need to be contract. So whatever calcium is present inside the muscle cell, the God kept this calcium into a storage area, and so that this calcium never directly goes to the sarcomere area and allows to muscle contraction. So what happens? First of all, our brain send the message via the nerve, and then this nerve reach into the muscle. Suppose this area need to be contract, so this nerve taking the signal, this reach over here in the form of action potential. So this action potential is present and coming from this area. This action potential enters into the T tubule, and this current or action potential activate the DHPR. Once the DHPR is activated, this is mechanically attached with the ryanodine receptor like a lock and key model, tala or chabi. And once this DHPR activate due to this action potential, guys, this will activate the ryanodine receptor, which is earlier was closed. Now this closed receptor will open up, and once it will be open up, guys, inside lots of calcium is there. This calcium starts moving outside, and this is going to be happen without using of the energy. We call it as a passive efflux of the calcium. Guys, this is due to the concentration gradient. Highs to low, this is moving. So this calcium going without any energy, it comes outside due to the opening of ryanodine receptor. It will goes into the sarcomere, reach into the troponin C area, activate the troponin, and this troponin remove the tropomyosin from the actin protein. Once the actin protein is now free with the tropomyosin, there is a myosin head which goes attached with this actin and leading to muscle contraction. So guys, this is how actually your muscle gets contract. Now please tell me, if your muscle is not getting any signal from the nerve, suppose your nerve get cut due to any fracture over here, what will be happen? The message can't go beyond this area, and message can't go beyond this area. What will be happening? This action potential won't be reached, DHPR won't be activated, and ryanodine won't open, no calcium efflux, no muscle contraction. So that is how the muscle gets paralyzed due to the nerve injury. Now please pay attention. Once the muscle contraction is done, now the muscle needs to get relaxed. Guys, if muscle needs to get relaxed, any free calcium which is not utilized, it need to send back into the sarcoplasmic endoplasmic reticulum. We need to send it back. And guys, now if this free calcium need to send back, guys, this is going from low side to higher side. Guys, we required a pump. And this pump we called as Sarca pump, which stands for sarcoplasmic endoplasmic reticulum calcium ATPase pump. This pump send this free calcium back into this storage house, and this is how the all calcium will be eliminated from this area, helps to the muscle relax condition. So this is how your muscle is actually contract. So if in a quick summary, if you will see that how muscle gets contracted, guys, the number one, there is a excitation via the action potential, which is simple part we called as a current. इसको क्या बोलेंगे बेटा? We called as a current. Then this excitation is happens with the help of nerve, and nerve release the neurotransmitter, and the neurotransmitter at neuromuscular junction is acetylcholine. This acetylcholine having a nicotinic receptor on this muscle area, which lead to the sodium influx, and on the end plate potential the muscle action potential will be generated. This action potential leading to excitation-contraction coupling. In this, there is an action potential spread along all the sarcolemma or the membrane of the sarcomere, enters into the T-tubule, activate the DHPR, and this leading to the opening of voltage-gated L-type calcium channel in T-tubule. Now, this act as a voltage sensor, and then it will activate the ryanodine receptor. This ryanodine receptor will be open up, calcium start releasing out from the sarcoplasmic reticulum, and DHPR mechanically activates the ryanodine receptor. Guys, please pay attention. Once this calcium start coming out from the sarcoplasmic reticulum, this goes to the troponin C, binds with the troponin C, remove the tropomyosin, and expose the actin binding area for the myosin. And myosin head binds with the actin, leading to using of this ATP. There is a power stroke, and this leads to the muscle contraction. ATP binds, detachment, ATP hydrolysis, and recocking of the myosin head. Guys, please pay attention. This ATP is required for detachment and relaxation of the muscle. So in the both condition, the ATP is required. And during the relaxation, calcium pumped back by the sarcoplasmic reticulum via the Sarca pump, and this calcium dissociate from the troponin C. Tropomyosin will come back, covers the actin, muscle gets relaxed. Guys, please pay attention. Whenever our muscle gets contract, जब भी अपनी muscle contract होती है ना बेटा, तो all the time this muscle contraction produce the heat. So guys, that is the condition when in the cold climate area, when we are going outside, guys, outside temperature is very low, our body get cold, and in this cold condition, our body maintain the homeostasis, the balance of the body, and so that we start shivering, and this shivering condition produce or generate the heat, and our core body temperature is actually increase. And this is how the muscle contraction, always remember that muscle contraction always generates the heat. Guys, please pay attention. If suppose this ryanodine receptor, which ideally should remain close all the time until unless no message coming from the nerve, guys, if suppose this ryanodine receptor get overactivated or remain open without any message of the nerve or the boss, guys, what will be happen? Muscle continuously gets contract, continuously heat generation will be there, and guys, this condition we called as malignant hyperthermia. What we call this? Malignant hyperthermia. So I hope now each and everyone is now easily understand what is the sarcomere and how this muscle is actually gets contract. Get it, guys? So I hope all you are enjoyed this session to understand of this sarcomere and the skeletal muscle condition. Right, guys? So most of the exams, sometimes they used to ask you that the step to the muscle contraction, sometimes they used to ask the different areas into the sarcomere. So please remember all of this part. Thank you for watching this video.